We continue in Chapter 2, Chemistry Comes to Life, talking about biological macromolecules. Biological macromolecules are the giant molecules of life. These are long chains called polymers made of repeating units called monomers. When polymers are made, water is removed and the reaction is called a dehydration synthesis. Oppositely, when the same molecules are broken apart, water is added and the reaction is called hydrolysis. This shows an example that two monomers are already bonded to one another and the third monomer to be added through dehydration synthesis. So polymers are formed by dehydration synthesis. So it's synthesizing or making a polymer. A water molecule is removed and two monomers are joined. Dehydration would be the water removal. Conversely, these polymers are broken down to monomers by hydrolysis. This is the addition of a water molecule. This disrupts the bonds between these monomers, producing taking a polymer and reducing it to a smaller, simpler compound of a monomer. Carbohydrates are polymers made of monosaccharides. Carbohydrates are composed only of carbon, hydrogen, and oxygen. Glucose and fructose are examples of monosaccharides or simple sugars, whereas sucrose and lactose are examples of disaccharides or double sugars. This image is shown in your textbook where you take a monosaccharide of glucose and a monosaccharide of fructose to make a disaccharide such as sucrose. Polysaccharides are chains of monosaccharides. These store energy and provide structure. The storage polysaccharide in plants is starch. In animals it is glycogen, which in humans we store primarily in the cells of the liver and muscles. Cellulose is a common example of an indigestible polysaccharide made from plants that consist of repeating units of glucose. Humans lack the enzyme necessary to digest cellulose, although cellulose is an important form of dietary fiber in the human diet. So as humans, we cannot digest cellulose because we do not have the enzyme necessary to break this down, but it is a part of our dietary fiber in our diet. Lipids are water-insoluble molecules made of carbon, hydrogen, and oxygen. These also store long-term energy, as well as they provide protection for vital organs and form the components of our cell membranes. Lipids are responsible for insulation. They also provide a means of absorbing lipid-soluble vitamins from the intestines, as well as transporting them to the cells of the body. Fats and oils are examples of triglycerides. These are polymers made of one molecule of glycerol and three fatty acids. Three fatty acids bond to glycerol through dehydration synthesis. If there are double bonds between the carbon atoms and fatty acids, triglycerides are classified as unsaturated. So if there's double bonds, they're unsaturated. If they contain the maximum number of hydrogen atoms, they are called saturated fats. Saturated fatty acids are so named because they are saturated with hydrogen ions. Fats and oils store about twice the energy per gram as carbohydrates or proteins. And so this shows an example of the glycerol to a fatty acid. That's what a triglyceride is. It contains one unsaturated fatty acid. It notes the triglyceride is a glycerol with 3 fatty acids. You can see in the example under B that you have an unsaturated fatty acid because you see the presence of the double bond between the carbon atoms. If you look over at this straight, these are considered saturated fatty acids. They do not curve. They would be in linear position because they contain the maximum number of hydrogen ions, atoms. They would not have any double bonds or triple bonds in them. Phospholipid molecules have a glycerol head that is polar and hydrophilic. They also have a fatty acid tail that's nonpolar and hydrophobic. Phospholipids are the primary components of the plasma membranes. The hydrophilic heads mix with the watery environments inside the cell and outside the cell, or intracellular and extracellular, whereas the hydrophobic tails point inward towards each other and help hold the membrane together. This shows the hydrophilic head, the water-loving, versus the hydrophobic tail, which hates water. So you see an image of the phospholipid membrane surrounding cells. So on the extracellular fluids, such as towards blood, you have the hydrophilic water-loving head. You have the hydrophobic tail facing inward. Intracellularly, or towards the cytoplasm, again, you have the hydrophilic water-loving head with the hydrophobic tail facing in towards the other hydrophobic tail from the extracellular fluid. Steroids are a type of lipid that consists of 4 carbon rings attached to molecules which vary from one steroid to the next. Estrogen and testosterone are examples of steroids, as is cholesterol. Cholesterol is a risk factor for heart disease when found in high levels in the blood. Steroids act as regulatory lipids that control metabolic processes or cellular functions. Proteins, amino acids, are the building blocks of proteins. These amino acids that form proteins are linked by bonds called peptide bonds. Peptide bonds are also formed through dehydration synthesis. Chains of only a few amino acids are termed peptides, whereas chains of 10 or more amino acids are called polypeptides. Proteins, polypeptide chains of at least 50 amino acids that provide structure. They also provide transport, and they also provide movement for the body. Proteins have 4 distinct levels of structure that affect their function. The primary, which is just the sequence of particular amino acids. The secondary level is whether it's in beta sheets or helixes, such as these will be formed by bonds and coils resulting from hydrogen bonding. Tertiary level is hydrogen bonds and disulfide bonds, which help to stabilize the shape of a protein. And quaternary consists of 2 or more polypeptide chains. So the primary sequence of particular amino acids are going to be determined by the genes. The beta sheets or helixes, such as the double helix in DNA, is going to be formed by bends and coils resulting from hydrogen bonding. Tertiary are going to be hydrogen bonds and disulfide bonds, which are going to be disulfide or much stronger bonds. These stabilize the shape of a protein, and the quaternary consists of 2 or more polypeptide chains. So the primary structure is the specific sequence of amino acid. These are going to be determined by the genes. The secondary structure, such as the helix in DNA, results from bending and coiling of the chain of these amino acids. Tertiary structure is the 3-dimensional shape, such as from the hydrogen and the disulfide bonding, stabilizing the protein. And then the quaternary structure results from the attractive forces between 2 or more subunits. Changes in the chemical environment of a protein can cause it to lose its structure, resulting in a loss of function. This is called denaturation. Factors that can affect a protein to denature are pH or heat, such as a high fever. Our proteins are responsible for regulating specific processes, such as in our skeletal muscle cell, proteins such as tropomyosin binding calcium, which we will study in later. So proteins control the processes that go on within our body. Enzymes are almost always proteins. These typically end in ASE. Enzymes speed up chemical reactions without being consumed. Without enzymes, chemical actions within our cells would occur far too slowly for it to sustain life. Metabolism is the term given to the chemical reactions that occur in the cell and involve energy transformation. Enzymes are very specific in their activity. This being specific is due to the unique shape of each enzyme's active site, such as individuals who are lactose intolerant. That's because they lack the enzyme lactase to break down the lactose. Acetylcholine is the neurotransmitter in skeletal muscle cells. The enzyme for acetylcholine is acetylcholine esterase. Enzymes factor 10,000 to 1 million times faster for these processes in our bodies to occur than would occur if there were not these enzymes to speed up these reactions. Enzymes bind to substrates at the active site. This forms an enzyme substrate complex. Sometimes there's cofactors, often termed coenzymes, that bind at the active site to facilitate the reaction. The substrate is converted to one or more products. So this shows the enzyme plus the substrate forms the enzyme substrate complex, yielding the products of the enzyme plus the product. You notice that the enzyme begins in the reaction and is also at the end of the reaction. So enzymes are not consumed in the process. They just act to lower the activation energy to speed reactions to occur. So in step 1, cycle begins when the active site of the enzyme is unoccupied and the substrate is present. The substrate binds to the active site of the enzyme, forming the enzyme substrate complex. The substrate is converted to products that are released from the active site, and the cycle can begin again. You see again the enzyme is released. Nucleic acids or genes or segments of polymers called deoxyribonucleic acid or DNA. DNA and ribonucleic acid, RNA, are the 2 types of nucleic acids that we will be talking about throughout the semester. Both are polymers of smaller units called nucleotides. So nucleotides are the building blocks for nucleic acids. A nucleotide is made up of a 5-carbon sugar bonded to one of 5 different nitrogen-containing bases and a phosphate group. The sequencing of the nucleotides in DNA and RNA determines the sequence of amino acids in proteins. The parental genetic material in the form of DNA provides our body with instructions for making specific proteins. RNA and DNA have structural differences related to the sugar, the bases, and the number of strands. RNA is single-stranded. It contains the sugar ribose, and it has the nitrogen-containing bases adenine, guanine, cytosine, and uracil. So this shows the ribose sugar, a phosphate. It contains cytosine, represented by the C, adenine, represented by the A, guanine by the G, and U by the uracil. DNA, on the other hand, has 2 strands that form a distinctive double helix. It contains the sugar deoxyribose and has the nitrogen-containing bases adenine, guanine, cytosine, and thymine. Uracil and thymine different. DNA has thymine, whereas RNA has uracil. This shows the double helix. It has the base pairing rules. G will pair with C, T with A. You can see the double helix, the sugar deoxyribose with the phosphate and one of the nitrogen-containing bases, either adenine, thymine, cytosine, or guanine. There's a special nucleotide called adenosine triphosphate, or better characterized, that we will be studying in the form of ATP. ATP is a molecule capable of storing energy in its phosphate to phosphate bonds. All energy from the breakdown of molecules such as glucose is channeled through ATP for the body to be able to use it. ATP is often described as the energy currency of the cell. And this shows adenosine triphosphate, where these broken bonds, these phosphate to phosphate bonds, release energy. When energy is released, this adenosine triphosphate becomes adenosine diphosphate, meaning that it only has 2 phosphate. So once it gains another phosphate, it's ATP, adenosine triphosphate, and that is the energy currency.